Good morning all. I'm Gurjit Singh, PhD candidate in Dr. Kensroy Lab, and today I'm going to present on autophagy in freezing wood frog model Rana sylvatica. So why do we study wood frogs and what promises do they hold? So wood frogs are one of the coolest animals in the animal kingdom, which are been extensively studied to understand the concepts of vertebrate freeze tolerance. They undergo whole body freezing during winter and become ice solid with no brain activity, no heartbeat, and no blood flow, but amazingly comes back to life during spring at 5 degrees unharmed without any major changes inside their body. So one of the major adaptation why they are able to do this is they produce huge amount of glucose, which is good enough to give us a diabetic shock. And there are many different pro-survival pathways which do get activated during this stress. So one of the pro-survival pathways which gets activated is autophagy. Autophagy is involved in maintaining the cellular homeostasis during different stresses. And interestingly, since wood frog faces most of these stresses during the freeze-thaw cycle during in wild and amazingly survives back when they thaw. So we were interested to understand if there is a basal level of autophagy involved in maintaining cellular homeostasis in different tissues. So we had three different tissues: liver, heart, and muscle. The conditions were controlled frogs at 5 degrees Celsius. We have frozen frogs at 24-hour freezing, and then we have recovered or 8-hour thaw frogs. So liver and heart are the main organs during freezing. They showed a heightened autophagic response during freeze-thaw cycle, which links to their role in maintaining other mechanism inside the cell during freezing. Muscle, it's one of the first organ to freeze when the freeze bout starts, and it's not the most metabolic active tissue. It shows overall low or no change in autophagic response. Overall, this study emphasized on tissue-specific role of this pro-survival pathway as well as interplay between different mechanisms inside the cell in the wood